All right. Thank you all very much. I'd like to turn it over to Michael Schatz from Johns Hopkins University, who's going to do a demo of Watershed.
Okay. Thank you very much. It's great to be here. Uh, this is actually kind of a really special place for me. I started in genomics 25 years ago, around the corner at what was the Institute for Genomic Research. And I was just like very fascinated for like how DNA impacts me and impacts all of us. And it's just a real pleasure to stand before you and sort of show off some of the latest advances, really taking advantage of interoperability and AI to make really great progress. And as you'll see in a second, now we have some really sophisticated models that can make really informed analysis to find variants of clinical significance, which personally is one of the main drivers of my research program. So the title is a mouthful, but the headline is we have this awesome new machine learning model called Watershed. It's now widely available for basically all the platforms, and we're open for business, and we want people to start using this. So I'm here to represent what I would consider a dream team of investigators. So there's myself. I'm here representing the AnVIL platform. I'm partnered by Elisa Manning from the Broad, representing Biodata Catalyst. We're also partnered with two excellent investigators, Stephen Montgomery and Alexis Battle, who are world's experts at genetics and the clinical interpretation, especially of something called rare variation. As I mentioned, one of my main pursuits is understanding sort of the implications of variation on disease. And this is a long-standing program in human genetics and beyond. And it turns out there's a spectrum there. So some variants in my genome and your genomes are quite common. They're shared by many, many, many people around the world. Although, as we kind of look at these variants, most of the time they have sort of marginal clinical impact. There are a few famous exceptions to this. There are some risk variants for, say, prostate cancer that are quite common but have relatively small risk, maybe 10%, 20%, 30% increase in risk. There are other variants that are more rare that occur in maybe, 1% of the population, 0.5% of the population. In the limit, there may be variants that only occur in one person, maybe me, maybe you, that are extremely rare. And it turns out as we kind of look across the survey of clinical phenotypes, those rare variants tend to be incredibly important, are incredibly impactful. They have these very important effect size, where not only are they increasing the risk of disease by some percentage of small percentage, it could be a 10-fold, 100-fold, 1,000-fold increase for risk. Certain variants, if you have them, you are more or less going to, with close to 100% chance, develop some condition. So it's really, really important to be able to study those. The challenge is they tend to be quite rare, and some of the statistical frameworks they're going to use to identify them are totally different. For common variants, things like GWAS are well established, but for rare variants, the statistics just break down. We need an entirely new approach. So that's where this project comes in. We need an entirely new approach, and we need tons of data. Well, what does NCPI have? Well, we have an entirely new approach, and we have access to tons of data and tons of compute, where we can kind of take advantage of all these resources to make progress on this clinical frontier here. So our project here is going to combine some of the data resources from the AnVIL, especially this really excellent project called GTEx, which has DNA and RNA sequencing data from about 1,000 individuals across 50 different tissues. This is, I would say, the premier resource for understanding human genetic variation and how it impacts gene expression. And then from the Biodata Catalyst side, we have excellent cohorts that have particular disease focuses associated with heart, lung, and blood disorders. So what we're going to do is marry these data resources together, bring together some of the compute capability, some of the workflow capability, some of the interactive capability, to hopefully be able to make progress on this rare disease analysis. So we had many goals here in terms of being able to stress test our interoperability in terms of data, workflows, interactive tools, users. And then ultimately, yes, we're going to, develop workspaces and GitHub repos and workflows. But ultimately, I want to have this have impact in the world. So we're going to demonstrate that by sort of analyzing some clinical cohorts, get those published into the world. And then hopefully, beyond anything that, that our team can do, we want the rest of the world to use this too. This is a community resource to really hopefully advance on clinical interpretation quite broadly. The core of this is something called the probabilistic graphical model. This is a machine learning technique. At a high level, it resembles a neural network in the sense that there's many inputs. They get aggregated together in this mathematical framework, and then we can make a series of predictions. By neural network standards, this would be a tiny network. But what drives this is something called a conditional random field, which is a very sophisticated mathematical modeling technique where we can integrate in all these weak signals. So at the top here, we can take genomic variations and annotations about those. So these annotations are things like, is this common? Is this encoding sequence? Is it a known regulatory sequence? Is it conserved in humans? Is it conserved in primates? Is it known enhancers? All, basically as many annotations as we can possibly get. Those become our input features. They get passed through to this special latent variable, which I'll explain in a second, that we're going to call here Z. And then we're going to have another readout, which is expression. This is our molecular phenotype that we're going to be able to use to be able to assess, are these variants important? Ultimately, what we want to do is make a prediction of yes or no. Is this variant that we just observed a functional regulatory variant? Now, the challenge for that is there are very few precious training examples. So you cannot set this up as like a standard supervised learning approach where you have a big matrix of, oh yeah, here's 1 million known variants of important regulatory function. No, these are extremely rare variants. They're very hard to identify. They're largely unknown. So we need an entirely new mathematical framework. And I really credit Alexis Battle and Stephen Montgomery, who invented this machine learning framework to be able to integrate these signals in this really nice framework to make accurate predictions on this. So it's a really powerful network. So the method that was published in 2020 is excellent for the analysis of single nucleotide variants. So this is when, say an A is flipped into a C, but it was blind to other types of variation. In particular, it was blind to structural variations. And in the recent few years, these are proving to be incredibly important for many, many disorders and diseases. They are, I would say, underexplored because at a technical level, they're very difficult to measure. These are things like insertions, deletions, inversions, translocations, things that we know happen in, say, a cancer context. Well, it turns out they also occur in what we think is normal, healthy tissue. They're just very difficult to get a readout on. But thanks to some new high-power long-read sequencing, it's now becoming a lot more powerful and efficient to have accurate recall of these structural variants. So the idea is we're going to take that existing watershed model and we're going to turbocharge it so that it can be appropriate for structural variants as well. And to do so, there's a lot of sort of research that went into this. I really credit Tanner Jensen, who's a student at Stanford, and Bohan Ni, who's a PhD student at Johns Hopkins, for leading this initiative to really kind of develop, well, what are the right set of features, what are the right set of annotations that can be applied in this mathematical framework, this probabilistic graphical model, to make accurate AI predictions on the importance of these variants. So this work was published earlier this year in Genome Research. In the publication, we describe how we train it in GTEx. We benchmark it against other tools. We also apply it in that time to a relatively small cohort of a few dozen samples from the Undiagnosed Disease Network, the UDN, a Common Fund project. And I'm very proud to say through this, we got a lot of new clinical insight into several of these cases. The UDN is really special in the sense that all these patients have already been evaluated through short-read sequencing, and they didn't find very much. That's sort of the recruitment strategy there. But now, revisiting those samples with long-read sequencing, powered by our new AI model, we could find what are variants that are that we're very confident. In fact, our clinical genetics collaborators have really basically validated that, yes, these are the variants that were associated with these developmental disorders. So we're really excited. We have this model. It's working great. In the last few months here, we've been making great efforts to make this accessible beyond our own research labs. We want to make it accessible to the whole world. And I really credit Bohan at Hopkins, and then also Paul Hansen, who's an analyst in Elisa's lab at Broad, who's really spearheaded the engineering effort to make this accessible. Okay, so what I'm going to try to do in the next 5 minutes here is give you a little demo. I do have massive imposter syndrome. As a PI, I'm pretty good at PowerPoint. But I really lean heavily on the students and engineers in my lab to actually do this. So let's give this a go. So the high level is I'm going to look for some interesting data. I'm going to look for the workflow that we want to run. And then I'm going to drop into AnVIL or Biodata Catalyst. In fairness, I'm an AnVIL expert, so I'm going to do AnVIL for this. But you could do equivalent operations in Biodata Catalyst as well. So bear with me here as I jump in. Okay, first up, I'm going to drop into the AnVIL portal here, anvilproject.org. I'm going to go look for the data that I'm interested in, hop over to the Explorer. And then the data set I'm going to use for demonstration purposes, of course, I'm not going to be using patient data or anything like that. I'm going to be using an open access data set called MAGE. This is gene expression data that was collected from gene expression data with matched DNA data from 731 individuals of diverse, globally diverse genetic ancestry. I will say in another NCPI project, that was the funding that supported the ingestion of this data set into this. This is another win for NCPI that now we have this great data set. These samples come from the so-called 1,000 Genomes project, which are consented for open release. This is an excellent resource for demonstrations. It's also an excellent resource for science. These are real measurements from these real people, well, real cell lines, and it's has now matched DNA and RNA data. I think of it as like a mini GTEx, but open access, which is great data sets available. So I found it in 2 seconds, so I can click export and we can send it to one of the portals here. I'm going to be using it inside of AnVIL just because, again, I'm here representing AnVIL in addition to NCPI. Okay, so now I have the data. The next step is, well, I want to do some analysis of it. I'm going to go to our friend DocStore, which is our kind of nice sort of repository for workflows where we can kind of go look for different tooling. I know, I happen to know the name of this is Watershed. But maybe you would search on some of the other keywords like rare variants or structural variants or other sort of terms there. And we can pop in and right there, again, this is all totally public. I happen to be signed in here, but there's nothing special about my account accessing this. So in here, there's a sort of a brief description about what the inputs are required. There are several, and I'll explain where those come from in a second. I wanted to highlight, we're also using a number of GA4 standards here. There's something called the Tool Registry Service, TERS. And then, so if you want to be able to pull this into other platforms, that's a great way to do this. The workflow itself is encoded as a WDL, a workflow description language that sort of encodes all the models and everything. So that's all available there. That's open in the public. And then thanks to our friends at DocStore, I really like these buttons over here. Ooh, launch with DNA Nexus, Terra, ELISA, AnVIL, or Biodata Catalyst. I would love to get a few more buttons here at the top of the list, Chris. The CFDE cloud workspace. Let's get that in. Okay, so we can go pop this in. Again, I can pick whatever platform we want to go to. I have it preloaded here. I'm gonna hop over to AnVIL. I guess I'm a power user. I have 846 workspaces in my account. So let's go look for Watershed. So I have several of these in my own, but importantly now we've made a public workspace where any of you can sign in. You can go find this. And this is going to have the data organized. Again, we're using that open access MAGE data, so there's no sensitivities about privacy or patient data. That was very much by design so that we could do this demonstration and record it and broadcast it on YouTube as necessary. Okay, so here's the sort of landing page for all of this that sort of gives you, some descriptions about where it comes from, some of the information, kind of an overview of what's necessary here. All that sort of standard information is available there. And then again, critically, I've already preloaded this with all the data that we want. At the top here is our metadata. So these are all those annotations, like how common it is in the global population. There's something called the ABC model that measures, is this variant likely to be an enhancer? There's something called CAD, which is a really hard and well-established method to establish the deleterious of different variants. A key thing is there's many of these annotations, and they don't necessarily have to be independent. That's one of the virtues of a probabilistic graphical model and the conditional random field. If there's dependencies between these annotations, no problem. It knows how to integrate those together and make sort of actionable results there. So we have all those annotation data. I also have sort of preloaded in all of the sample data. Again, 731 samples, globally diverse. This is the mixture of DNA, RNA data kind of mixed and matched in here. Notably, all of the data are accessible via DIRS. So this goes to data.taro.bio. So if you're on any of the platforms that know how to speak DIRS, we can go ingest them into your platform as well. So we have the workflow, we have the annotations we need, we have all the sample data that we need. The next step will be then to flip into the analysis that we want to do here. And there are a few steps. Some of them are kind of, if you've ever done anything in bioinformatics, step one is always clean up your data. Make sure it's sort of in the right shape and format and everything that you need to do. There's some normalization that needs to take place. So there are a few steps there. There's notebooks associated with all those steps to kind of get things into place there. The next thing we really want to do, though, is then, once things are organized, we want to click off the Watershed WDL, the actual workflow that does the heavy lifting about making all these predictions here. A really awesome part of this is like if you have, I don't know, 1 sample, 100 samples, or 100,000 samples, it's basically equally easy to run at any scale, right? That's the virtue of being on all these platforms. We can do this at massive scales, depending on how large your sort of clinical cohort is that's available to you. So I'm not going to do it now because it will take a few minutes, but again, all these annotations are loaded, all of the DNA data, all the RNA data get sort of processed in, and then we can click off the analyze button, launch button, and then this will kick off this workflow in the backend. Here I'm going to do a little sort of movie magic here and then advance to where the results are already deposited. So that's in the run table here. And then again, there's sort of information, the kind of the key column. There's a lot of columns to kind of keep things organized, but the key column is going to be this output. OP. Yeah, output that has this combined data set. Now, as great bioinformatics people like to do, the raw output is very raw. It's this giant table where for every structural variant that you have, what is its sort of association with every gene, and there's a bunch of metadata that's associated there. So if you wanted to follow up with more information about different scores, about different genes, that's all available, of course. But of course, it's also useful to kind of get an overview of what happened. So in addition, there are these sort of nice sort of post-processing workflows and notebooks that kind of summarize the results for you. So here, this is available. Again, you can just click on it. We can scroll through a bunch of analysis. The key thing I want to highlight at the end, which is a summary table about how important these different rare variants and these rare structural variants are with respect to gene expression outliers. So the x-axis here is a measure of like how extreme the expression is. This is measured in a measure of z-score standard deviations. So if you're 5 standard deviations away from normal, there is almost always a structural variant nearby. That's sort of the interpretation of this plot. And then if you want to go dive in and go into investigate, which SVs are driving that, that's where that sort of raw table exists. So again, hopefully this is sort of a rapid fire demonstration about just how powerful it is to bring forth all of these data to be able to make all these clinical predictions associated with it. Okay, in the slide deck, I have screenshots in case you want to review at home. But then just to wrap up, so the main model has been published, demonstrating, again, in GTEx UDN. We're actually presenting this and a few updates at ASHG in a couple weeks. The workflow and the workspace are now available in DocStore, and then in any of the Terra platforms, you can go get a look at the workspace. We're writing a separate paper about the cloudification, where we're going to demonstrate it on 1,000 genomes, and I think we're going to tap into some of the top med cohorts there as well. And then we're like days away from clicking the big run to be able to launch this on the scale of thousands of samples from top med, where we have matched DNA and RNA data. So I'm that's in the context of NCPI. We're also applying this internally to other clinical samples we have at Hopkins and beyond. If you've seen earlier versions of this talk, there were plans for other efforts using things like the T genome and the new HPRC. I think we're still pushing forward on those, but we're looking for other creative funding mechanisms for that. So that, I'll thank you. Happy to take any questions. But again, I'm just super excited about NCPI and all the accomplishments that we've had here over the last several years. Thanks, everybody. Please. Many. At ASHG, we're going to be presenting a refinement. So this is the published model is really good at analyzing structural variants that are following in regulatory sequence. We did notice when the structural variants overlap the coding sequence, there's some special considerations that need to be happening there, so we're taking care of those. I think we're going to make also refined models that are working in like pedigrees, where you have sort of extra information about transmission and other things. I will say another initiative that is going on now and I think a lot of groups are interested in this, is our training foundation models that can make sort of a variety of multi-omics predictions and clinical predictions, based out of genome sequencing data. So there's a whole ensemble. To a first approximation, it's like all AI all the time to advance on these fronts.
My next one is, considering the amount of data that you're using to train this and use it, are you concerned about model failure? And how are you addressing that? And my next question after that is going to be, and sorry, in terms of the users.
Yeah.
What type of resources are provided to help the users understand more about how the limitations of these types of models that are being used and these tools are being used, so that they can be better prepared and know what to expect when they're running them?
Great questions. There's a lot of sort of harmonization that has to occur first. Throughout these, in the interest of time, I'm going to go through it, but throughout these notebooks, there's a bunch of like QC metrics that are reported, and we also say, well, if you see this, this can be a red flag of something is being off. So we do try to help you with that. But I'm with you, right? I mean, I think this has been one of the headlines from the last several years is like, maybe you develop, I don't know, GWAS or a pathogenic risk score in one population doesn't necessarily transfer very well to other populations. So that's something that we're very interested in, and we have other kind of projects there. The question I thought you were going to ask me, which you just reminded me to think about, was I thought you were going to ask me, well, how much does this cost? And the good news is once you have the variance called and once you have the expression quantified, it's like $1 for 1,000 samples. Incredibly inexpensive. So once we have that heavy lifting done, actually doing the model fitting is quite rapid and inexpensive.
Apologies if this is too simple a question, but you found these structural variants. How would you characterize the strength of that evidence then, and where would you contribute those findings? I'm thinking about, my variant info, or, how would somebody else find that evidence that you found? How would they refer back to it?
So the
the
model itself makes a probabilistic assignment that says, yes, with some confidence, I think this is a variant that is important, and sort of here's the metrics for it. So at the level of individual variants, that's important. But I think you're asking kind of a broader question about like, once Mike finds this variant that's important, how would other people then go discover it?
Yeah.
I think this is a challenge in the field. There are certain databases, ClinVar and friends and Nomad and friends that are really excellent at single nucleotide variants. But just like the nuts and bolts encoding and structural variants, like kind of breaks down. So this is where I'm very excited about things like Verse, where you can kind of have a more robust representation of those variations.
Yeah.
Okay. All right. Well, thanks everybody.